Humans are animals. You should probably know that already. So let's talk about what animals are and all of the different features we have and what features we share with different groups of animals. Animals are pretty cool, and there are a lot of them. These are probably the organisms you're most familiar with, and I hope you have a favorite. But let's have some context. So here we're back looking at our phylogenetic tree of life. Here we have the three domains, bacteria, archaea, and eukarya. We, of course, are eukaryotes, and we are right there, Kingdom Animalia. So what does it mean to be an animal? Do you know the features we share? I mean, there are a lot of different animals out there, and really the only commonalities that all of us have are that we're multicellular, we have multiple cells, and we're heterotrophs. Remember, hetero means different, troph means eat. So this means we have to eat something other than ourselves. Most organisms are heterotrophs. Plants that can make their own food, those are really the exception. But let's look at some of these really beautiful and interesting animals out there. Here are three examples. We have Phylum Porifera, Phylum Cnidaria, and Phylum Ctenophora. These are all beautiful things, and they look a little bit different than maybe some of the animals you're used to, but these are all animals, and yes, including the sponge. This phylogenetic tree is nice because it actually overlays the different features that go along these different branches. So you can see all the way at the bottom we have multicellularity. All animals are multicellular, and we share this because that first animal ancestor was multicellular. Well, we can see here at this first branch off point, sponges, Phylum Porifera, they don't have true tissues, but everyone else does. So let's look a little bit more. So here in our pineapple under the sea, Phylum Porifera are multicellular, but again, they don't have any tissues. You can also see they don't really have any symmetry. These are just kind of random globs. They might, they tend to be in tubes, or at least some of them, but there isn't true organization. But let's look at what these tissue layers are that all other animals have. We have our ectoderm on the outside. These cells turn into our skin, hair, and nails. In the middle, we have our mesoderm. Meso, so of course middle. These are our bones, the muscles, our connective tissue, a couple parts of our brain. Really, this is the bulk of us, at least. And in the middle, we have our endoderm. Endo means within. And this is our gut and a few organs that are associated with our gut. And most animals have these three tissue layers. You can see it here in our worm, but we have them as well. But let's go back to our tree here. So we've talked about our tissues. The next thing we want to talk about is types of symmetry. So now we're seeing our Radiata, they have radial symmetry, and then everyone else has bilateral symmetry. So what is this Radiata? Here we have two phyla. We have Phylum Cnidaria, and then we have Phylum Ctenophora. Cnidaria, those are the jellyfish that you are probably already familiar with. Phylum Ctenophora, they are similar, but they are a little bit different. These are our box jellyfish, and these are the ones you really want to watch out for. Box jellyfish, if they sting you, that's bad. Most jellyfish, some will be painful, but most are actually pretty okay. But what these guys have is they have this radial symmetry. So you can see they have a center axis and everything is symmetrical from that axis. We, on the other hand, we have bilateral symmetry. So you can put a line down the center and the two halves are almost exactly symmetrical. But let's go back. The next thing we want to talk about is the body cavity. So now we have a couple different ways we can have a body cavity. So you can see at the top we have something called an acoelomate. It doesn't have a coelom, and here a coelom is a fancy word for body cavity. So our flatworms, they do not have that cavity. They are simply a gut, a mass of tissue, and then their skin. Now in the middle we have our pseudocoelomate. So we have their gut in the middle. There is a space between their gut or endoderm and their mesoderm. And we find these in roundworms. A coelomate, we are one of these. So now we have our endoderm. It's encased in a little bit of mesoderm, but now there's a hole through that mesoderm. And this is what a true coelom is. So the next thing we want to talk about is we have different types of development. So different groups here will develop in different ways. These two ways are protostomes or deuterostomes. You can see it has to deal with how once the embryo gets a certain number of cells, it is a empty ball, and then that ball invaginates, folds in on itself to form those three tissue layers. And how it does that determines whether it's a protostome or a deuterostome. Protostomes are insects, crustaceans, snails, and octopoda, and deuterostomes are starfish and vertebrates. You can see what's really going on here is whether that first hole where it does that invagination becomes the mouth or the anus first. So protostomes are mouth first, deuterostomes are anus first, and you can see some humans still demonstrate this this capability. Next, now we want to talk about notochords and segmented appendages. We're getting closer and closer to what we are. Now we're talking about hard-bodied organisms, so we're starting to get to some of these more interesting things that you're probably familiar with. If we look back at some of these very first fossil animals, these are all soft and squishy, and these are the ancestors that we're talking about all those traits we just discussed. Unfortunately, they don't fossilize well, and they're not quite as exciting. But here in the Cambrian, that's where we first get these hard-bodied animals, and this is what we call the Cambrian explosion. The Cambrian is one of my personal favorite times in the fossil record because we just get so many interesting and weird animals. And one of the ways this is exciting is all of a sudden we have a lot more fossil life than we did before. There's a couple things going on. First, these are the first instances of hard-bodied animals, and we think this is the first instance of a true predator-prey relationship. Animals have finally figured out that they can eat other animals. So they are hard-bodied because they're trying to protect themselves from each other, and then predators are trying to be bigger and badder so they can eat everyone else. And in this period, we also see the ancestors of a lot of the major phyla that are alive today. So we have some examples here. We have Cutanascolix barbarensis, then we have Opabinia regalis. Here's some other genera we can see. So we have Opabinia, Hallucigenia, Picaya, Morella, and Ischia. Hallucigenia is one of my favorites because it just looks like this horrible spike monster that's, the thing of nightmares. The Cambrian explosion just has so many new and interesting forms that are so unlike any of the animals we see today. Evolution was really experimenting and just trying all sorts of new things. Not all of them worked, but this is the beauty of evolution. We just get to try things and then we see what survives. And, well, a lot of them don't. But one of the cool things is we see the ancestors of Phylum Chordata, to which we belong. Phylum Chordata, all of these animals have a notochord or a dorsal nerve cord. So they have a nerve running the length of their body. We also see some pharyngeal gill slits, and now we have a post-anal tail. So that means the anus isn't all the way at the end of the organism like it was in most of our worms. These are tunicates or sea squirts. They are in Phylum Chordata. They look really weird, but they are related to us. I love this one here that looks like this kind of bizarre heart with purple veins. And these guys are filter feeders, so their embryo looks kind of like that tiny little minnow thing, but they actually grow up to be like this. So once they become adult, they are sessile. They just stick in one spot on the ocean floor and filter feed from there. But now we can look at a tree looking of all of our deuterostomes. So here we have the origin of our notochord, and you can see a couple other traits mapped onto this tree, such as a head and vertebral column. Our tunicates are right there, or Urochordata. The next thing we want to talk about is Subphylum vertebrata. So now we are talking about our vertebral column or spine. One pedantic note, it is not a spinal column because it is a column of vertebra. We do have that spine, the nerve cord going in the middle, but we don't have a column of spine. One of the fun things here, though, is we just have an explosion of different animals within subphylum vertebrata. So this was a very successful strategy because we're encasing that important nerve cord within bone to keep it nice and safe. We see the first vertebrates in the Cambrian, actually. We have this Milocunmingia. It's not quite as exciting as the rest of the animals we see in the Cambrian, but it is one of our very earliest ancestors. Lampreys are a modern fish that are some of the simplest living vertebrates. So you can see they're just kind of this little fishy thing with a dorsal nerve cord and a vertebral column encasing that dorsal nerve cord. But the next period within the fossil record we want to talk about is the Devonian. The Devonian is known as the age of fishes. So like the Precambrian, we saw this explosion of lots of weird life. In the Devonian, we see the explosion of bony, armored fishes. So we have a lot of different fish in the ocean. And the fact that all of them had this bony armor meant they were probably all eating each other. And there's a lot of predator-prey relationships going on. But let's talk about vertebrates and why you might want to be a vertebrate. Some of the interesting characteristics they have are lifelong motility. All of them are mobile throughout their entire lives. So unlike that tunicate or sea squirt that is sessile for its adult life. Also, they are larger in body size. Other animals might be smaller, especially all those worms. And we have this internal bony skeleton. So we are supported on the inside by the bones, and that's why we can be so tall and just not be a squishy little blob on the floor. We also see a cranium. So we're seeing the concentration of all of these sensory organs and a brain in one spot at the front of the organism. And that's where we concentrate all of those special senses. We also start to see an endocrine system, or a system where different organs can communicate with each other through hormones. We also see neural crest cells, or the beginnings of immune systems, and we also have a closed circulatory system. So rather than blood just kind of being everywhere, we actually have a specific system with which to transport blood and other molecules throughout the body. And now we're also seeing specific red blood cells. But let's look at a tree of different vertebrates and how they are all related to each other. So here are cartilaginous fishes. Those are sharks. Our ray-finned fishes. Those are most of the fish that you're probably familiar with. But let's talk about these guys, our Sarcopterygii. Sarcopterygii is a fancy name for lobe-finned fish. So you can see in our picture here, ray-finned fishes, they do not have bones in those fins on the side. It still works fine for fins, but our lobed fin fishes, they have stronger fins because there are bones in those fins. What's kind of fun is we actually thought these guys were extinct. We discovered the fossils before we realized that there were still living versions of these guys. So these are coelacanths. They just live super, super deep. So it was a while before we encountered a living one. This is a fun picture because you can see how many millions of years ago if you go left to right. But you can also see about how deep these different organisms lived. So you can see the coelacanth, the modern day lobe-finned fish, is pretty deep. Then we have Eusthenopteran and Pandorictes and Acanthostega that are still living in the water. But then these two at the top, Tiktaalik and Ichthyostega, these guys are beginning to move on land. So it's through these lobe-finned fishes that animals were finally able to make the jump to land because they had enough reinforcement into their front fins to be actually able to move on land. Let's take a closer look here. So Eusthenopteran, he was a fossil lobe-finned fish, but still definitely living in the water. Ichthyostega, this is a clear tetrapod, one of the earliest here. So he's definitely living on the land. And a really exciting fossil find was Tiktaalik. So Tiktaalik is right there in the middle. So this was a transitional structure because there are more bones in their front fins, but not clearly formed limbs yet. So we were all really excited when Tiktaalik was found. But let's talk about us, superclass Tetrapoda. So tetrapoda just means 4 feet. And this is really all land animals. You might want to might make a note that not all tetrapods actually have 4 feet. Like birds, they have 4 limbs, just 2 of them happen to be wings and the other 2 are feet. And we are also tetrapods, even though we have 2 hands and 2 feet. We also still consider snakes tetrapods, even though they don't actually have any limbs. They have simply lost those limbs secondarily. But let's talk about our first tetrapods. We have Acanthostega in the Devonian, and then we have Eriops, slightly younger in the Permian. So you can see they're very amphibian or kind of crocodile-like eyes still at the top of their head. They might be living in shallow water, wanting to look above them. The things that we find in tetrapods is now all of these guys have thicker skin. They can walk on land, and now we're starting to see differentiated limb muscles. We also see a stronger skeleton because now we don't have water to kind of support us a little bit. And now we have the development of lungs. We are losing our gill slits because we are breathing on land rather than in the water. We have eyelids because we need to protect our eyes from drying out. We also have enhanced hearing. This is a fun one because sound actually propagates more easily through water than through air. So that's why if you live on land, you actually need to try a little bit harder to hear what's going on because sound just doesn't propagate as well. So that's why we see slightly more developed hearing in all of our land-dwelling animals. So here's a phylogenetic tree of all of our tetrapods. But of course, this is a class about humans. So let's zone in on our mammals. You can also see there's a bunch of other things going on with all of our other tetrapods. They are just as interesting, just as derived, and just as exciting. We just don't tend to emphasize them when we're talking directly about humans. But these first mammals, they first evolved in the Triassic, so they were coexisting with the dinosaurs here. They were just very tiny and, kind of rat-like and honestly unexciting at this point. One thing that we want to talk about when we're talking about the fossil record is there are at different points in time, different animals roam or rule the earth. And what allows this to happen are these things, extinctions. When we have a mass extinction, we see a turnover. All of the animals after that extinction are completely different. And a particular extinction, the asteroid that killed off the dinosaurs in the Cretaceous-Tertiary boundary around 65 million years ago, this is what allowed mammals to become dominant because it killed off all of those dinosaurs that were previously occupying all of those ecological niches. This is particularly exciting for geology because we have this asteroid that hit the Earth. It let a layer of dust pretty much all over the Earth. So if you know what you're looking for, this is one of the easiest things to find when you're looking at a geological rock strata. So you can see here's some different people pointing out that KT boundary. And this is fun because it's relatively easy to figure out if a fossil is before or after the KT boundary because it does tend to be so clear in geological deposits. Let's talk a little bit about mammals and what we are. So here we're comparing a mammal cranium to a reptile cranium. And now you can see our mandible or our jawbone is changed a little bit. Instead of having multiple bones in our jaw, we've actually moved multiple of those bones up into our ear, and now we just have our dentary bone, which we now call our mandible. And overall, this just creates a strengthening of a jaw. There's fewer moving parts, and this is actually the densest bone in our body. So we have a little bit more chewing power or feeding efficiency when we're comparing mammals to reptiles. There are, of course, several other traits we have. We have mammary glands, hair. We are homeotherms, or we can generate our own body heat, unlike most reptiles. We also have heterodonty. Remember, hetero means different, donty is teeth. So we have specific different types of teeth. We have incisors, canines, premolars, and molars. What's interesting is pretty much all other reptiles and fish just kind of have lots of the same teeth. That allows them to replace their teeth more easily, but now we can have specialized functions for each of our teeth. We also have our limbs are oriented under our body, so this is much more efficient for running or quick movement than reptiles where their legs still kind of stick out to the side there. And mammals have been supremely successful. There are so many different types of mammals. We could talk on and on about all the different groups of mammals here, but let's zone in on our primate relatives. So we are most closely related to colugos or sugar gliders. Some people occasionally call them flying lemurs, but remember they aren't lemurs and they glide, not fly, so that's one of the not great names. But we're also relatively closely related to rodents and rabbits. They form a clade called glaries. Tree shrews, colugos, and primates together, this is U. arconta, and all of us together are U. arconta glaries. Let's remind ourselves of the important traits that we find in primates. We have a postorbital bar, we have an opposable thumb, but also prehensile fingers. We're very good at grabbing things. Our brain is a little bit more complex. We have nails instead of claws. We can delicately grasp things. Our eyes face forward, and overall, we have a relatively short snout. We just talked about a lot. When you're trying to make sense of all of this, it really helps to find something that you think is exciting or cool and use that as a way to ground this and figure out how this all fits together. I recommend trying to find a phylogenetic tree, look at how different things are related to each other, and also figure out what's your favorite and learn more about your favorite fossil organism. Mine personally is the Cambrian explosion and all of the weird animals that existed there, but remember, find your favorite.